This is Farmer Joe. It was just an ordinary day in 1879 where he was working in the fields when he noticed something odd about his tobacco plants. And to his surprise, the leaves of his plants were mottling and curling. So out of curiosity, he asked his scientist friend Adolf Meyer, an agricultural director from the Netherlands, to take a look at his plants. Meyer had no idea what was wrong with Joe's plants, so he did some experimentation to see if there was a difference between the dead and healthy leaves. He experimented with temperature, fertilization, and light to see if any of these factors affected the plants. He noticed that when the sap from the dead leaves came in contact with the healthy ones, they became infected. To find the cause of this infection, he tried to reproduce the disease. He implanted the infected sap into bacteria and animal feces. Unfortunately, Adolf Meyer wasn't able to find the agent of this disease, so he asked his friend Dmitry Ivanovsky, who was a virologist, to see if he could figure it out. He prepared a solution with the infectious agent and put it through a fancy filter known as a Chamberlain filter. This filter works by allowing fluids to pass through small pores while keeping bacteria and other microorganisms contained. People at this time used this filter to purify water. He was surprised to find that after the solution was filtered, it was still capable of infecting more plants. Although he knew this meant that the agent was much smaller than a bacteria, he still did not know what the infectious agent actually was. A couple of decades later, in 1898, microbiologist Martinus Behring had to step in to solve this mystery. He discovered that the infectious agent had to be a virus. This was considered a mind-blowing discovery at the time, since most scientists believed that infectious diseases were caused by germs. Meyer, Ivanovsky, and Behring's efforts using filtration allowed for the science community to now analyze the structures of viruses in later years. Now that we know how the tobacco mosaic virus was discovered, let's take a look at how it works on a microscopic level. The tobacco mosaic virus, also known as TMV, spreads mechanically through human contact or infected plants to unaffected plant leaves that are damaged. The TMV virus has a rod-like structure and is composed of 2,130 copies of coat proteins, also known as protein shells, that encase the virus's RNA molecule. RNA is a single-stranded chain of cells that processes proteins. The molecular process that it undergoes begins with the virus entering damaged areas of plant leaves. The virus's particles begin to disassemble, exposing the TMV's RNA and forming a positive sense RNA. A positive sense RNA is essentially composed of viral messenger RNA that directly translates into proteins. The RNA in the virus then begins the translation process a few minutes after the virus infects the plants. The RNA rod ends then bind to the replicase, which is an enzyme that works as a catalyst to start the synthesis of an RNA molecule that uses an RNA template. In the TMV virus, a negative sense RNA is produced to serve as a template for the production of the positive sense RNAs, which are then translated by host ribosomes to produce coat proteins. The coat proteins then proceed to assemble the progeny virions, which are essentially the children of the parent virus. This causes the formation of extremely stable TMV particles, which causes plant cell damage as well as the physical appearance of curled and dried up leaves on infected plants. Now that Farmer Joe knows how the TMV virus works on a microscopic level, he got his hands on a modern computer and he researched how this crazy disease spread in his fields. Since the demand for tobacco increased, he planted more fields of tobacco crops and very close together, which unfortunately allowed the virus to spread from plant to plant. He also realized that he did not clean his farming tools often enough because the disease came from contaminated tools. He also yelled at his workers for smoking tobacco near the plants, because when they touched the plants, they may have transferred the disease to them. Farmer Joe also finally understood why the disease was called tobacco mosaic virus, as the most common symptom was the dark and light discoloration of the leaf, which creates a mosaic appearance. Other symptoms are necrosis, which is the death of the organs and cells inside of the living organism, stunting the plant's growth, leaf curling, and yellowing of plant tissues. Although these symptoms are all very common with the infection of this virus, the age and environmental factors that the plants are in play a great factor in how these symptoms affect the organism. This virus is wreaking havoc on more than just Farmer Joe's plants. Over the last century, the tobacco mosaic virus has had far-reaching global consequences. Because tobacco is in high demand, the countries that produce it have seen economic success. However, as crop production increased, the virus's spread also increased significantly. This resulted in a 30 to 35% decrease in tobacco yield and a 50% decrease in gross value. Early mosaic infection of tobacco plants have been shown to cause a 60% reduction in crop value. In addition to the devastating effects of virus transmission, it has had a negative impact on the economy, resulting in a $5.7 million annual global loss. This demonstrates that the virus is more than just a plant virus. It is also an economic virus. Just like Farmer Joe, you may be asking yourself, how can we reduce the spread of this disease? The most common method of eliminating tobacco mosaic virus is through the practice of proper sanitation. A worker must wash their hands when handling the tobacco plants. If someone finds infected plants, they should immediately remove them from the field in order to avoid the disease from spreading to the soil beds of the plants. Farmers should avoid smoking near these crops to prevent the chances of tobacco mosaic virus getting in contact with the plants. It's been noted through research that greenhouses are breeding grounds for the virus due to having temperatures around 40 degrees Celsius. Farmers should rethink about growing their plants in enclosed areas such as greenhouses. Finally, a cross protection method can be used to make plants more resistant to this disease. A mild strain of TMV can be placed in the plant to prevent a more harmful strain from attacking the plant. It's almost like a vaccine. With these sanitary methods, the tobacco industry can thrive, and farmers can enjoy their TMV-free tobacco.